Today we'll talk about the epistasis. What is epistasis? This epistasis, epi means above, upon, and stasis means standing. So epistasis is a gene interaction in which one gene suppresses or inhibits the expression of the other gene. So we can say that in epistasis it is just a gene interaction. One gene suppresses or it inhibits the expression of the other gene. What is epistasis? In epistasis, it is just a gene interaction in which one gene, it masks, it inhibits, it suppresses the expression of the other gene. That is called as epistasis. This the gene, for example, we have this condition. This is A, and this A, it inhibits the expression of B gene. It inhibits the expression of B gene. That means A gene is epistatic gene. The gene which inhibits the expression of other gene is known as epistatic gene. That means in this case A gene inhibits the expression of B gene. That is why it is called as epistatic gene. And the gene whose expression will get suppressed or whose expression will get masked, that is called as hypostatic gene. Or it can be like this. This A gene, it inhibits the expression of B gene. So this is what is called as epistatic. This A gene, it is called as epistatic gene, and B is called as, it inhibits the expression of A gene, inhibits the expression of B gene. That is called as epistasis. So the gene which inhibits the expression of other gene is called as epistatic gene or inhibiting gene. Epistatic gene is also called as inhibiting gene. And the gene whose expression will get inhibited, that is called as hypostatic gene. This epistasis, it is similar to dominance recessiveness, but in case of epistasis, two genes occupy different gene loci. In case of epistasis, two genes occupy different gene loci. For example, we'll see in case of dominance and recessiveness. This is, for example, this is a case of dominance recessiveness. This or smaller. This dominant allele, it inhibits the expression of small, or recessive allele. Dominant allele inhibits the expression of recessive allele. It is simply a dominance recessive relationship. It is dominance recessive relationship. This epistasis is similar to dominance recessive relationship, but in case of epistasis, two genes occupy two different gene loci, whereas in case of dominance, two alleles of the same gene, they occupy the same gene locus. That means dominant allele inhibits the expression of the recessive allele. Capital R inhibits the expression of the small r. In case of epistasis, what happens? this two genes occupy different gene loci. For example, this A, it inhibits the expression of this A, it inhibits the expression of B. That is, A is epistatic gene or inhibiting gene, and B is hypostatic gene. That means these two genes occupy the two different gene loci. This is one gene loci, this is another gene loci. That means epistatic gene is at one gene loci and hypostatic gene is at another gene loci. It can be like this also. We have A, and then hypostatic gene can be present on other chromosomes. This is this is A gene. This is present at one gene locus, and hypostatic gene B, this is present at another gene locus. So here A is epistatic gene, and B is hypostatic gene. They occupy the two different gene loci. Another important thing is that this dominance recessive relationship, it is just an allelic relationship, whereas in case of this epistasis, it is a non-allelic relationship. It works at non-allelic level. These are non-alleles. This A gene and B gene, they are non-alleles, whereas capital R and small r, these are alleles. So this works, dominance recessiveness works at allelic level, and epistasis works at non-allelic level. If we'll discuss about what is the difference between dominance and recessiveness, and epistasis, we'll talk about the dominance and epistasis. This dominance and epistasis, this dominance, it works at intragenic level. That means only one gene is involved. That is, only one gene is involved in dominance recessiveness. Whereas it works at intergenic level. That means different genes are involved. This is one gene, this is another gene. Different genes are involved here. It is an allelic interaction. These are two alleles of the same gene. Whereas epistasis, it is a non-allelic interaction. It is non-allelic. This in case of dominance recessive relationship, only the dominant allele is able to inhibit the recessive allele. That means only dominant allele can inhibit the expression of recessive allele. Only dominant allele can inhibit the expression of recessive allele. That means this is a dominant allele and this is a recessive allele. Only R gene can inhibit the expression of small r. Small r, this recessive allele cannot inhibit the expression of R, capital R. Whereas in case of epistasis, dominant non-allele as well as recessive non-allele can inhibit the expression of the dominant non-allele as well as recessive non-allele. can inhibit the expression of can inhibit the expression of other gene. That means, see, we have condition here. We have this A and A. This is B and B. This A, this is dominant allele. It can inhibit the expression of B. So this is called as when the dominant allele will inhibit the expression of other gene. That is called as dominant epistasis. Here, the dominant allele of a gene, it inhibits the expression of other gene. That is called as dominant epistasis. But a recessive allele can also inhibit the expression of other gene. So it would be like this. We have small a, and this is B and B. Here, this is homozygous recessive condition. These are recessive alleles. They inhibit the expression of B gene. So this is called as recessive epistasis. So in case of dominant epistasis, the dominant allele inhibits the expression of other gene, whereas in case of recessive epistasis, the recessive allele inhibits the expression of other gene. So here, in case of dominance recessive relationship, only the dominant allele inhibits the expression of. That means here we have dominant allele or small, or it is recessive allele. This only or, capital R, can inhibit the expression of small r. Small r cannot inhibit the expression of r. But in case of recessiveness, dominant allele as well as recessive allele can inhibit the expression of other gene. So this was regarding the difference of the. Then we have another difference. In case of dominance recessiveness, that means the expression of only recessive allele is suppressed. That means the expression of only recessive allele is suppressed. Expression of only recessive allele is masked. So here, expression of only this allele, recessive allele, is suppressed or inhibited. While as in case of epistasis, expression of both dominant allele as well as recessive allele can be suppressed. So it would be like this. So here it can be small b, it can be small b here also. It means that this will explain like this. That means this is a dominant epistasis. The expression of dominant allele as well as recessive allele both can be suppressed. For example, instead of this capital B, here also can be this small b. So we can say that expression of both dominant allele as well as recessive allele can be inhibited. Expression of both dominant as well as dominant or recessive allele, recessive, precisely non-allele, can be inhibited. So we can say that in case of dominance recessiveness, only the expression of recessive allele is inhibited, whereas in case of epistasis, the expression of both dominant non-allele as well as recessive non-allele can be inhibited. For example, here we have dominant allele, dominant non-allele, it is expression is inhibited. Here we have dominant recessive non-allele, here its expression is suppressed. So this epistasis, another important thing regarding the epistasis, that it reduces the number of epistasis reduces the number of phenotypes in F2 because of the suppression or inhibition activity of the epistatic gene. So epistasis, it reduces the number of phenotypes in F2. That means this epistasis, it reduces the number of phenotypes in F2 because of the suppression or inhibition. This is due to the inhibition effect of epistatic gene. As a result of that, this normal dihybrid ratio, it gets modified in case of epistasis. It gets modified to 12:3:1, or it gets modified to 9:3:4, and this 12:3:1, it is in case of dominant epistasis. It is in case of dominant epistasis, and 9:3:4, it is in case of recessive epistasis. This epistasis, it reduces the number of phenotypes in F2 because of the suppression activity or inhibition activity of the epistatic gene. As a result of that, the normal dihybrid ratio, that is 9:3:1, that gets modified to 12:3:1 or 9:3:4, and this 12:3:1 we get in case of dominant epistasis, and this 9:3:4 we get in case of recessive epistasis. So what is dominant epistasis? In case of dominant epistasis, the dominant allele inhibits the expression of the other gene, whereas in case of recessive epistasis, this recessive allele it inhibits the expression of the other gene, that is called as recessive epistasis. So here we have seen that this here the dominant allele inhibits the expression of other gene, this is called as dominant epistasis, and this recessive epistasis, that means recessive allele inhibits the expression of other gene, that is called as recessive epistasis. So we have epistasis. Epistasis is of two types. One is called as dominant epistasis, and recessive epistasis. In case of dominant epistasis, the ratio is 12:3:1, and in case of recessive epistasis, the ratio is 9:3:4. In case of dominant epistasis, we have this condition. The dominant allele inhibits the expression of the other gene, The dominant allele, this is dominant allele, it inhibits the expression of the other gene. And in case of recessive epistasis, we have small a, capital B. This recessive allele inhibits the expression of the other gene. That means this small a inhibits the expression of the other gene. This is called as recessive epistasis. In recessive epistasis, the recessive allele inhibits the expression of other gene, and in case of dominant epistasis, the dominant allele inhibits the expression of the other gene. And in case of recessive epistasis, we get 9:3:4 ratio, and in case of dominant epistasis, we get 12:3:1 ratio. This was regarding the what is epistasis. Thanks.